Oh hey, I didn't see you there. You remember that last time in my last video I said about creating the world's first reindeer unicorn? Next time on Professor Patrick's lab, we will find out how I made the world's first reindeer unicorn from the Chernobyl. Until next time, yeehaw! Come on, yeah! That's right, and today I'm gonna show you how you could create your very own DIY reindeer unicorn at home using this science called molecular genetics. Welcome back to Dr. Professor Patrick's Secret Science Lab, out here in a very secret location, where today I'm here with my assistant, Cherie. Yeah, let's go. All living cells contain genes, which are the units of inheritance. Now these genes are basically the blueprints of the cell, telling them how they should function, how they should grow within a tissue, and these genes should have the ability to be copied perfectly so that their daughter cells will be identical and there will be no defects. Now these genes are made of DNA, which my partner now, Cherie, will explain to you.
So the genetic molecule consists of DNA. So DNA stands for deoxyribonucleic acid, while RNA stands for ribonucleic acid. So like all polysaccharides and proteins, DNA and RNA are all macromolecules, or also called polymers. So these polymers, they are made up of a lot of monomers joined together in a very long chain. So the monomers of DNA and RNA are nucleotides, and therefore DNA and RNA are called polynucleotides, or also called nucleic acids. So this is the basic structure of a nucleotide. So it consists of a phosphate group, a pentose sugar, and also a nitrogen-containing base. So this is the structural formula of a phosphate group, and the sugar can be either deoxyribose or ribose sugar. So the difference between the deoxyribose and the ribose sugar is that the deoxyribose sugar, they have fewer oxygen than the ribose sugar. And then there are also different nitrogen-containing bases, which are called adenine, guanine, thymine, and cytosine for DNA. But for RNA, it consists all of these except for thymine. However, it contains uracil instead. So each of these bases will have their own respective pairs. So adenine will bind with thymine, while guanine will bind with cytosine. So they will form a different number of hydrogen bonds. So adenine and thymine will form 2 hydrogen bonds, while guanine and cytosine will form 3 hydrogen bonds. So each phosphate molecule will bind with a pentose sugar, which will form a large chain. And when it zooms out, it will form some kind of a staircase, where the sugar phosphate acts like a support for the ladder, while the base pairs will act as the steps. So this staircase will then spiral up and will form a double helix structure known as the DNA.
DNA has the unique ability to make copies of itself. Now this process is called DNA replication. First, an enzyme called helicase unbinds the parental DNA. The DNA unzips itself by breaking the weak hydrogen bond in between the base pairs and forming 2 template strands, starting with the 5' to 3' and starting from the 3' to 5'. And they have exposed nucleotides.
So the place where the helicase attaches itself and unzips the DNA is called the replication fork.
And helicase may attach to different parts of the DNA strand to make replication bubbles. which in turn makes replication faster.
After this happens, single-strand binding proteins attach and stabilize the two separate parental strands. The 5' to 3' will become the leading strand, and the 3' to 5' will become the lagging strand. DNA polymerase will then attach to the 3' end of the 5' to 3' parental strand. The DNA polymerase attaches free nucleotides to the complementary nucleotide on the parental DNA. However, since DNA polymerase can only add nucleotides to the 3' end of the parental strand 5' to 3' must be replicated in fragments that must later be joined together. This only happens in the lagging strand. This is where RNA polymerase or primase comes in. RNA primer is attached to a segment of the strand by the primase. The short primer fragment is then extended by DNA polymerase, and transcription now continues in the 5' to 3' direction, forming an Okazaki fragment. After the RNA primer is replaced by DNA polymerase as shown previously, the enzyme DNA ligase joins the Okazaki fragments to the growing strand. Two new identical DNA daughter strands are now formed, completing a replication bubble.
Now my friends, I'm sorry to say, but that's all the time we have left for today. I hope you learned something useful from our brilliant lesson here. I know that you are so impatient and you're so eager to create your very own reindeer unicorn at home. But don't worry, our next episode will be out soon. Please stay tuned and come back. We'll see you next time in Dr. Professor Patrick's Secret Science Lab.